Chapter 8 Cell, the Unit of Life. The physicochemical approach to study and understand living organisms is called reductionist biology. Reductionist biology. Reductionist biology. Dash discovered the triple helical structure of collagen. G.N. Ramachandran. G.N. Ramachandran. G.N. Ramachandran. The two contributions of Ramachandran in biology are discovery of the triple helical structure of collagen, analysis of the allowed conformations of proteins through use of Ramachandran plot. Discovery of the triple helical structure of collagen, analysis of the allowed conformations of proteins through the use of Ramachandran plot. Discovery of the triple helical structure of collagen, analysis of the allowed conformations of proteins through the use of Ramachandran plot. Dash first saw and described a live cell. Anton von Leeuwenhoek. Anton von Leeuwenhoek. Anton von Leeuwenhoek. Cell was first seen by Robert Hooke. Robert Hooke. Robert Hooke. Dash discovered the nucleus. Robert Brown. Robert Brown. Robert Brown. Matthias Creeden was a German or British botanist or zoologist. German botanist. German botanist. German botanist. Theodor Kohn, a British or German botanist or zoologist. British zoologist. British zoologist. British zoologist. Scladen examined plants and observed that all plants are composed of different kinds of cells in year 1838. 1838. 1838. Swan studied different types of animal cells in year 1839. 1839. 1839. Dash reported that cells has a thin outer layer which is totally known as plasma membrane. Swan. 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 Scladen concluded that presence of cell wall is a unique character of plant cells. False. Swan concluded this. False. Swan concluded this. False. Swan concluded this. Dash proposed that the bodies of animals and plants are composed of cells and products of cells. Swan. 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 Cell theory was given by Scladen and Swan. Scladen and Swan. Scladen and Swan. Dash first explained that new cells are formed from pre-existing cells. Rudolf Virchow in 1855. Rudolf Virchow in 1855. Rudolf Virchow in 1855. Omnicellula E. cellula was given by Rudolf Virchow. Rudolf Virchow. Rudolf Virchow. Main arena of cellular activities in cell is cytoplasm. 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 Prokaryotes have membrane-bound organelles. False. 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 Microbodies are present in prokaryotes. False. 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 Ribosomes are non-membrane-bound organelles. True. 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 Ribosomes are found only in cytoplasm. False. 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 Organelles in which ribosomes are found: rough endoplasmic reticulum, chloroplast and mitochondria. 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 Two non-membrane bound organelles in animal cells are ribosome and centrosome. Ribosome and centrosome. Ribosome and centrosome. Centrosome helps in cell division. Cell division. Cell division. Ecoplasma are only dash in length, 0.3 micrometer. 0.3 micrometer. 0.3 micrometer. Generally, bacterial length is 3 to 5 micrometer. 3 to 5 micrometer. 3 to 5 micrometer. The largest isolated single cell is ostrich egg. Ostrich egg. Ostrich egg. Diameter of human RBC 7 micrometer. 7 micrometer. 7 micrometer. Size of virus is 0.02 to 0.2 micrometer. 0.02 to 0.2 micrometer. 0.02 to 0.2 micrometer. Size of PPLO is 0.1 micrometer. 0.1 micrometer. 0.1 micrometer. Size of typical eukaryotic cell is 10 to 20 micrometer. 10 to 20 micrometer. 10 to 20 micrometer. The prokaryotic cells are represented by bacteria, blue-green algae, mycoplasma and PPLO. Bacteria, BGA, mycoplasma and PPLO. Bacteria, BGA, mycoplasma and PPLO. Eukaryotes multiply more rapidly than prokaryotes. False. Prokaryotes multiply more rapidly than eukaryotes. False. Prokaryotes multiply more rapidly. False. Prokaryotes multiply more rapidly. The four basic shapes of bacteria are bacillus rod-like, coccus spherical, vibrio comma-shaped, and spirillum spiral. Bacillus rod-like, coccus spherical, vibrio comma-shaped, and spirillum spiral. Bacillus rod-like, coccus spherical, vibrio comma-shaped, and spirillum spiral. Cell wall is present in all prokaryotes except mycoplasma. Mycoplasma. Prokaryotes have single chromosome. True. Prokaryotes have circular DNA. True. Small extrachromosomal DNA is called plasmid DNA. Plasmid DNA confers certain unique dash to bacteria. Phenotypic characters. Example of such a character is resistance to antibiotics. Dash is used to monitor bacterial transformation with foreign DNA. Plasmid DNA. Prokaryotes have something unique in the form of inclusions. Specialized differentiated form of cell membrane called dash is the characteristic of prokaryotes. Mesosome. Prokaryotes have a chemically complex cell envelope. True. The three layers of cell envelope are outermost glycocalyx, cell wall, plasma membrane. Outermost glycocalyx, cell wall, plasma membrane. Staining procedure developed by dash divide bacteria into categories, namely staining procedure developed by gram divide bacteria into categories such as gram positive and gram negative. By gram positive and gram negative. By gram positive and gram negative. Two-sheath glycocalyx is called slime layer. Thickened of glycocalyx is called capsule. Cell wall function. Determine shape of the cell and provide strong structural support. Prokaryotic cell membrane is structurally similar to eukaryote. True. Extensions of plasma membrane in the cell are three forms. Name them. Vesicles, tubules, and lamellae. Mesosome help in cell wall formation, DNA replication, DNA distribution to daughter cells, respiration, secretion, increase surface area of the plasma membrane, increase enzymatic content. Cell wall formation, DNA replication, DNA distribution to daughter cells, respiration, secretion, increase surface area of the plasma membrane, increase enzymatic content. Cell wall formation, DNA replication, DNA distribution to daughter cells, respiration, secretion, increase surface area of the plasma membrane, increase enzymatic content. In cyanobacteria, there are other membranous extensions into cytoplasm called dash, which contain dash. Chromatophores, pigments. In cyanobacteria, there are other membranous extensions into cytoplasm called chromatophores that contains pigment. Bacterial flagella, three parts are filament, hook, and basal body. Longest portion out of the three is filament. Cilia is absent in bacteria. True. Dash are elongated tubular structure made of a special protein. Pili. Dash are small bristles like fibers sprouting out of the cell. Fimbriae. Pili and fimbriae also play a role in motility. False. Ribosome size 15 nanometer by 20 nanometer. 15 nanometer by 20 nanometer. Ribosome subunits in prokaryotes are 50s and 30s. Ribosome attach to single mRNA and form a chain called dash or dash. Polyribosomes are polysome. The ribosome of a polysome translates the mRNA into proteins. True. 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 Reserve material is stored as inclusion body. Inclusion body. Inclusion body. Inclusion bodies are bounded by membrane. False. 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 Example of inclusion bodies: phosphate granules, cyanophagin granules, glycogen granules. Phosphate granules, cyanophagin granules, glycogen granules. Phosphate granules, cyanophagin granules, glycogen granules. Gas vehicle are found in blue, green, purple and green photosynthetic bacteria. Blue, green, purple and green photosynthetic bacteria. Blue, green, purple and green photosynthetic bacteria. Identify the organelles in the plant cell shown in diagram 1. Diagram 1A: rough endoplasmic reticulum, B nucleus, C nucleolus, D Golgi apparatus, E nuclear envelope, F plasma membrane, G vacuole, H middle lamellae, I cell wall, J mitochondria, K ribosomes, L chloroplast, M cytoplasm, N peroxisome, O microtubule, P plasmodesmata, Q smooth endoplasmic reticulum, R lysosome. A rough endoplasmic reticulum, B nucleus, C nucleolus, D Golgi apparatus, E nuclear envelope, F plasma membrane, G vacuole, H middle lamellae, I cell wall, J mitochondria, K ribosomes, L chloroplast, M cytoplasm, N peroxisome, O microtubule, P plasmodesmata, Q smooth endoplasmic reticulum, R lysosome. A rough endoplasmic reticulum, B nucleus, C nucleolus, D Golgi apparatus, E nuclear envelope, F plasma membrane, G vacuole, H middle lamellae, I cell wall, J mitochondrion, K ribosomes, L chloroplast, M cytoplasm, N peroxisome, O microtubule, P plasmodesmata, Q smooth endoplasmic reticulum, R lysosome. Identify the organelle in the animal cell shown in diagram 2. Diagram 2: A microvilli, B plasma membrane, A microvilli, B plasma membrane, C centriole, D peroxisome, E lysosome, F ribosome, E lysosome, F ribosome, G mitochondrion, H rough endoplasmic reticulum, G mitochondrion, H rough endoplasmic reticulum, I cytoplasm, J nucleus, I cytoplasm, J nucleus, K nucleolus, L nuclear envelope, M smooth endoplasmic reticulum, N Golgi apparatus. Extensive compartmentalization is not seen in eukaryotes. False. 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 Organelle present in animal cell but absent in plant cell is centriole. 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 Cell membrane 72. Detailed structure of membrane was studied only after the advent of dash electron microscope. Electron microscope. Electron microscope. Cell membrane are mainly composed of lipid and protein. Lipid and protein. Lipid and protein. Polar head of lipid is towards the outer or inner sides. Outer. 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 Lipid component mainly consists of phosphoglycerides. 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 In the lipid component, in addition to phospholipid, membrane also contains dash cholesterol. 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 Cell membrane don't possess carbohydrate. False. They possess carbohydrate. False. They also possess carbohydrates. False. They possess carbohydrate. Human erythrocyte has approximately -% protein and -% lipids. 52% proteins and 40% lipids. 52% protein, 40% lipids. 52% protein, 40% lipids. On the basis of dash, the types of membrane, two types of membrane proteins are ease of extraction, integral and peripheral protein. On the basis of ease of extraction, two types of membrane proteins are integral and peripheral protein. Ease of extraction, integral and peripheral protein. Ease of extraction, integral and peripheral protein. Peripheral proteins lie on the surface of membrane. True. 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 Integral proteins are always totally buried inside the membrane. False. Partially or totally. False. Partially or totally. False. Partially or totally. Which model of plasma membrane is shown in diagram 3 A to E? Fluid mosaic model of plasma membrane. A. Sugar. B. Peripheral protein. C. Phospholipid bilayer. D. Integral protein. E. Cholesterol. This is fluid mosaic model of plasma membrane. A. Sugar. B. Peripheral protein. C. Phospholipid bilayer. D. Integral protein. E. Cholesterol. Fluid mosaic model of plasma membrane. A. Sugar. B. Peripheral protein. C. Phospholipid bilayer. D. Integral protein. E. Cholesterol. Fluid mosaic model was given by whom and in which year? By Single and Nicholson in 1972. By Singer and Nicholson in 1972. By Singer and Nicholson in 1972. The quasi-fluid nature of lipid helps in lateral movement of protein. Lateral movement of protein. Lateral movement of protein. The ability to move within membrane is measured as its fluidity. 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 Fluid nature of membrane is also important because of function like cell growth, formation of intercellular junctions, secretion, endocytosis, cell division. Cell growth, formation of intercellular junction, secretion, endocytosis, cell division. Cell growth, formation of intercellular junction, secretion, endocytosis, cell division. Define osmosis. Movement of water by diffusion through a selectively permeable membrane is called osmosis. Movement of water by diffusion through a selectively permeable membrane is called osmosis. Movement of water by diffusion through a selectively permeable membrane is called osmosis. A membrane is selectively permeable to some molecules present on either side of it. True. 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 Example of active transport: sodium potassium pump. Sodium potassium pump. Sodium potassium pump. Cell wall is non-living. True. 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 Cell wall functions: give shape to cell, protect cell from mechanical damage and infection, help in cell to cell interaction, provide barrier to undesirable substances. Give shape to cell, protect cell from mechanical damage and infection, help cell to cell interaction, provide barrier to undesirable molecules. Give shape to cell, protect cell from mechanical damage and infection, helps in cell to cell interaction, provide barrier to undesirable molecules. Algae cell wall is made up of cellulose, galactans, mannans and minerals like calcium carbonate. Cellulose, galactans, mannans and minerals like calcium carbonate. Cellulose, galactans, mannans and minerals like calcium carbonate. In other plants, it consists of cellulose, hemicellulose, pectins and proteins. Cellulose, hemicellulose, pectins and proteins. Cellulose, hemicellulose, pectins and proteins. The primary wall is capable of growth. True. 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 The secondary wall is formed on the inner or outer side of the cell. Inner. 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 The middle lamella is a layer mainly made of calcium pectate. Calcium pectate. Calcium pectate. Function of middle lamellae: holds different neighboring cells together. Holds different neighboring cells together. Holds different neighboring cells together. Dash and dash may be transversed by dash which connect the cytoplasm of neighboring cells. Cell wall and middle lamellae, plasmodesmata. Cell wall and middle lamellae, plasmodesmata. Cell wall and middle lamellae, plasmodesmata. Endomembrane system. Each organelle of the endomembrane system is distinct in terms of its structure and function. True. 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 Endomembrane system includes Golgi complex, endoplasmic reticulum, lysosomes and vacuoles. Golgi complex, endoplasmic reticulum, lysosomes and vacuoles. Golgi complex, endoplasmic reticulum, lysosomes and vacuoles. Functions of mitochondria, chloroplasts and peroxisomes are not coordinated with endoplasmic reticulum. True. 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 Identify the organelles shown in diagram 4, label A to E. Endoplasmic reticulum. A. Nucleus. B. Nuclear pore. C. Rough endoplasmic reticulum. D. Ribosome. E. Smooth endoplasmic reticulum. Diagram 4 is endoplasmic reticulum. A. Nucleus. B. Nuclear pore. C. Rough endoplasmic reticulum. D. Ribosome. E. Smooth endoplasmic reticulum. Diagram 4 endoplasmic reticulum. A. Nucleus. B. Nuclear pore. C. Rough endoplasmic reticulum. D. Ribosome. E. Smooth endoplasmic reticulum. The endoplasmic reticulum divides the intracellular space into dash and dash compartments: luminal and extraluminal. Luminal and extraluminal. Luminal and extraluminal. Endoplasmic reticulum with ribosome is called rough endoplasmic reticulum. Rough endoplasmic reticulum. Rough endoplasmic reticulum. In the absence of ribosome, it is called smooth endoplasmic reticulum. Smooth endoplasmic reticulum. Smooth endoplasmic reticulum. RER is frequently observed in the cells actively involved in protein synthesis and secretion. Protein synthesis and secretion. Protein synthesis and secretion. RER is continuous with outer nuclear membrane. Outer nuclear membrane. Outer nuclear membrane. SER functions: synthesis of lipids and steroids, detoxification of drugs. Synthesis of lipids and steroids, detoxification of drugs. Synthesis of lipids and steroids, detoxification of drugs. Identify the organelles shown in diagram 5. Label X. 5 is Golgi apparatus. X is diagram 5 is Golgi apparatus. X on the X on the cistern. Diagram 5 Golgi apparatus X cistern. Golgi apparatus X cistern. Dash in here dash observed densely stained reticular structures near the nucleus. Golgi in 1898. Golgi in 1898. Golgi in 1898. Golgi bodies consist of dash shaped or dash disc sacs or cisternae. Disc shaped sacs or cisternae. Disc shaped sacs or cisternae. The sacs and cisternae are of dash micrometer diameter, 0.5 micrometer to 1 micrometer, 0.5 micrometer to 1 micrometer, 0.5 micrometer to 1 micrometer. They are stalked parallel or perpendicular to each other. Parallel. Varied number of cisternae are present in a Golgi complex. True. Forming phase is also called dash phase is concave or convex. Forming phase is also called cis phase, and that is convex. Also called convex. Forming phase is also called cis phase and also called convex phase. Forming cis convex. Dash phase also called dash phase is concave. Maturing phase also called trans phase is concave. Maturing phase also called trans phase is concave. Maturing trans concave. The cis and trans phase of the organelle are entirely different. True. Cis and trans phase are not interconnected. False. Golgi apparatus function: packaging, modification, transportation. Golgi apparatus deliver material only to intracellular targets. False. Both intracellular and outside targets. False. Both intracellular and outside targets. Materials are packaged in the form of dash vesicles. Materials fuse with dash phase of Golgi apparatus and emerge from dash phase. Fuse with cis phase and emerge with trans phase. Fuse with cis phase and emerge with trans phase. Fuse with cis phase and emerge from trans phase. Golgi apparatus is important site of formation of dash and dash. Glycoprotein and glycolipid. Lysosomes are not membrane bound. False. Lysosomes are membrane bound. Lysosomes are formed by the process of dash in dash. Formed by packaging in Golgi bodies. Lysosomes are rich in dash enzymes. Hydrolytic. Its enzymes are optimally active at acidic or basic pH. Acidic pH. Lysosomal enzymes can't digest nucleic acids. False. It can digest carbohydrate, protein, fats as well. False. It can digest carbohydrate, proteins and fat as well. False. It can digest carbohydrate, proteins, fats as well as the nucleic acids. Vacuole contain water, sap, excretory product and other non-useful material. Water, sap, excretory product, other non-useful materials. Water, sap, excretory product, other non-useful material. Vacuole membrane is called tonoplast. Tonoplast. In plant cells, vacuoles can occupy up to -% of the volume of the cell. 90%. 90%. Tonoplast functions facilitate transport of a number of ions and other materials against concentration gradient into vacuole. Facilitate transport of a number of ions and other materials against concentration gradient into vacuole. Facilitate transport of a number of ions and other material against concentration gradient into vacuole. Concentration of some ions is higher in cytoplasm than in vacuole. True. Contractile vacuole is present in dash and is important for amoeba for excretion and osmoregulation. Amoeba for excretion and osmoregulation. Food vacuole are present in protist. Identify the parts of mitochondria shown in diagram 6. Diagram 6. A. Outer membrane. B. Inner membrane. C. Matrix. D. Inner membrane space. E. Crista. A. Outer membrane. B. Inner membrane. C. Matrix. D. Intermembrane space. E. Crista. A. Outer membrane. B. Inner membrane. C. Matrix. D. Intermembrane space. E. Crista. Mitochondria are stained by dash dye. Janus green. The number of mitochondria per cell is always constant. False. It is dash shaped with diameter of dash micrometer, average dash micrometer. Sausage 0.2 to 1 micrometer, average 0.5 micrometer. Sausage 0.2 to 0.1 micrometer, average 0.5 micrometer. Sausage 0.2 to 1 micrometer, average 0.5 micrometer. Its length is dash micrometer. 4.1 micrometer. The inner compartment is filled with matrix. The inner membrane forms a number of enfoldings called cristae. Cristae function increase surface area. The two membranes have their own specific enzymes associated with mitochondrial function. True. Anaerobic respiration does not happen in mitochondria. True. RNA is absent in mitochondria. False. Mitochondria DNA is single or double and circular or linear. Single circular. Mitochondria multiply by fission. Plastids are found in all plant cells and dash. Euglenoids. Mitochondria is larger in size than plastids. False. 3 types of plastids are chloroplast, chromoplast. Identify the type of plastids shown in diagram 7, label A to F. Diagram 7, chloroplast. A. Outer membrane. B. Inner membrane. C. Granum. D. Thylakoid. E. Stroma lamellae. F. Stroma. Diagram 7 is chloroplast. A. Outer membrane. B. Inner membrane. C. Granum. D. Thylakoid. E. Stroma lamellae. F. Stroma. Diagram 7 is chloroplast. A. Outer membrane. B. Inner membrane. C. Granum. D. Thylakoid. E. Stroma lamellae. F. Stroma. Chloroplast contain pigments: chlorophyll, carotenoids. Chromoplast have dash soluble dash type of pigments like fat soluble pigments like carotenoid, example carotene xanthophyll. Fat soluble carotenoid, example keratin xanthophyll. Fat soluble carotenoid, example keratin xanthophyll. Dash gives the part of the plant a yellow, orange or red color. Chromoplast. Leucoplasts are colorful or colorless plastids. Tell their function. Colorless store nutrients. Potato contain which plastid? Amyloplast. Tell the three types of leucoplast and their function. Amyloplast store starch, oleoplast store oils and fats, aleuroplast store proteins. Amyloplast store starch, oleoplast store oils and fats, aleuroplast store proteins. Amyloplast store starch, oleoplast store oils and fats, aleuroplast store proteins. Majority of chloroplasts are found in dash cells. Mesophil. Length and width are length 5 to 10 micrometer and width 2 to 4 micrometer. Length 5 to 10 micrometer, width 2 to 4 micrometer. Length 5 to 10 micrometer, width 2 to 4 micrometer. Shapes of chloroplast are lens-shaped, oval, spherical, discoid or even ribbon-like. Lens-shaped, oval, spherical, discoid or even ribbon-like. Lens-shaped, oval, spherical, discoid or even ribbon-like. One chloroplast per cell is present in Chlamydomonas. Mesophil cells have dash chloroplast per cell. 20 to 40. Matrix in mitochondria is analogous to dash in chloroplast. Stroma. The outer membrane is relatively less permeable. False. Thylakoids arrange in stacks to form dash. Grana is also called dash. Intergranal thylakoids. Thylakoid of different grana are connected by stroma lamellae. The membrane of the thylakoids encloses a space called lumen. Stroma contains enzymes required for the synthesis of carbohydrate only. False. Carbohydrate and protein both. It contains single stranded DNA. False. Double stranded DNA is present. Double stranded DNA is present. DNA is circular or linear? Circular. Chlorophyll pigments are present in the stroma. False. It is present in thylakoids only. It is present in thylakoids only. Both chloroplasts and mitochondria have dash ribosomes. 70S ribosomes. Ribosome, cytoskeleton, cilia and flagella. Identify the organelles found in diagram 8 and its part A and B. 8 is ribosome, A large subunit, B small subunit. Ribosome, A large subunit, B small subunit. Ribosome, A large subunit, B small subunit. Ribosomes were observed by dash in dash year by George Faraday 1953. They are composed of dash and dash. Proteins and RNA. Here S is dash unit stands for dash coefficient. Schwarzbach's unit sedimentation coefficient. Schwarzbach's unit sedimentation coefficient. Schwarzbach's unit directly or indirectly measure dash and dash of ribosome. Indirectly, density and size of ribosome. Eukaryotic ribosomes are dash S, while the prokaryotic ribosomes are dash S. 80S and 70S. 70S ribosome subunits are dash and dash tell the types of rRNA in each subunit. 50S 30S contains 16S RNA and 50S contain 23S and 5S RNA. 50S 30S contain 16S rRNA and 50S contain 23S and 5S rRNA. 50S, 30S contain 16S rRNA, 50S contain 23S and 5S rRNA. 8S ribosome subunits are dash and dash. Tell the types of rRNA in each subunit. 60S and 40S. 40S contains 18S rRNA and 60S contains 28S, 5.8S, 5S rRNA. 60S and 40S. 40S contain 18S rRNA and 60S contain 28S, 5.8S, 5S rRNA. 60S and 40S. 40S contain 18S rRNA. 60S contains 28S and 5.8S, 5S rRNA. An elaborate network of filamentous proteinase structures present in cytoplasm is called cytoskeleton. Cytoskeleton consists of which three types of filamentous proteinase structures: microtubules, microfilaments, intermediate filaments. Microtubules, microfilaments, intermediate filaments. Microtubules, microfilaments, intermediate filaments. Cytoskeleton function: mechanical support, motility, maintenance of shape of the cell. Mechanical support, motility, maintenance of shape of the cell. Mechanical support, motility, maintenance of shape of the cell. A section cilia or flagella is shown in diagram 9. Identify its parts A to F. Diagram 9: A. Plasma membrane, B. Peripheral microtubules, doublets, C. Interdoublet bridge, D. Central microtubule, E. Radial spoke, F. Central sheath. Diagram 9: A. Plasma membrane, B. Peripheral microtubule doublets, C. Interdoublet bridge, D. Central microtubule, E. Radial spoke, F. Central sheath. A. Plasma membrane, B. Peripheral microtubules doublets, C. Interdoublet bridge, D. Central microtubule, E. Radial spoke, F. Central sheath. Dash and dash are hair-like outgrowths of cell membrane. Cilia and flagella. Cilia are small or large structures which work like dash, causing the movements of either the cell or the surrounding fluid. Small oar-like structure. Flagella function: cell movement. Prokaryotic and eukaryotic flagella are structurally similar. False, different. Cilia and flagella are not covered with plasma membrane. False, covered with plasma membrane. Their core is called axoneme. Axoneme possesses a number of microfilaments running parallel to the long axis. False, microtubules, not microfilaments. Axoneme have dash number of microtubules arranged laterally and dash number located in the center. 18 9 doublets 2. 18 9 doublets 2. Such arrangement is called 9 plus 1, as 9 pairs are laterally arranged and 1 pair is centrally located. False, 9 plus 2 as 9 pairs are located peripherally and 2 microtubule in center. False, 9 plus 2 as 9 pairs are located peripherally and 2 microtubule in center. False, 9 plus 2 as 9 pairs are located peripherally and 2 microtubules is in center. The central tubules are connected by dash and is enclosed by a dash. Bridges connected by central sheath. Central sheath is connected to one of the tubules of each peripheral doublets by a dash. Radial spoke. Total number of radial spokes in cilia are 9. The peripheral doublets are also interconnected by linkers. Interdoublet bridge. Both the cilium and flagellum emerge from dash structure called centriole basal bodies. Centriole Centrosome and centriole. A transverse section of centriole is shown in diagram 10. Identify which part A to F. A. Macule or pericentrioidal satellite. B. CA connective. C. Triplet fibril. D. Central rod hub E. 9 spokes. F. Cartwheel structure. A. Massive or pericentriolar satellite. B. C. A connective. C. Triplet fibril. D. Central rod hub. E. 9 spokes. F. Cartwheel structure. A. Massive or pericentriolar satellite. B. C. A connective. C. Triplet fibril. D. Central rod, that is hub. E. 9 spokes. F. Cartwheel structure. Centrosome contain 2 centrioles. Centrosome are surrounded by amorphous pericentriolar materials. Amorphous pericentriolar materials. Centrioles lie parallel or perpendicular to each other, which has an organization like a dash. Perpendicular like a cartwheel. Perpendicular like cartwheel. Arrangement is dash 9 plus 0. Centrioles have 9 even or uneven spaced peripheral fibrils of dash protein. Even tubulin protein. Each peripheral fibril is a doublet. False. Triplet. The adjacent triplets are not linked. False. They are linked. Central part of proximal region of centriole is called dash, which is connected with peripheral triplets by dash. Hub radial spokes. Hub and spokes are made of dash. Protein. Centrioles form basal body of cilia or flagella and spindle fibers. Basal body of cilia or flagella and spindle fibers. Total number of microtubules in centriole is 27 9 triplets. Nucleus was first described by dash in year dash. Robert Brown 1831. The material of nucleus was stained by acidic or basic dyes was given the name dash by dash. Basic chromatin by Fleming. Basic dye chromatin by Fleming. The interface nucleus has nucleoprotein fibers called dash. Chromatin. Identify the parts of nucleus shown in diagram 2. Diagram 2. A. Nucleoplasm. B. Nucleolus. A. Nucleoplasm. B. Nucleolus. A. Nucleoplasm. B. Nucleolus. The space between the two nuclear membrane is of dash nanometer and is called dash space. 10 to 50 nanometer and is called perinuclear space. 10 to 50 nanometer perinuclear space. 10 to 50 nanometer perinuclear space. The outer membrane remains continuous with endoplasmic reticulum. Nuclear pore formed by fusion of the two nuclear membranes. Movement of dash and dash takes place through nuclear pore. RNA and proteins. Nucleoplasm contains nucleolus and chromatin. Nucleoli are dash shaped structures. Spherical. Nucleoli are membrane bound. False. Active ribosomal RNA synthesis occur in dash. Nucleolus. Large and more numerous nuclei are present in cells actively carrying out protein synthesis. True. Chromatin contains non-histone proteins. True. RBC doesn't contain a nucleus, hence it is non-living. False. Chromatin contains DNA, histones, some non-histone proteins and RNA. DNA, histones, some non-histone proteins and RNA. Human DNA, that is diploid DNA, is approximately dash meter long. 2 meter, exactly 2.2 meter. 2 meter, exactly 2.2 meter long. 2 meter, exactly 2.2 meter long. Content of nucleolus is continuous with the rest of the nucleoplasm. True. 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 Identify the structure shown in diagram 12, label X. Diagram 12 is chromosome X kinetochore. Chromosome X kinetochore. Chromosome X is kinetochore. Primary construction is called dash, on the side of which disc-shaped structures dash are present. Centromere kinetochores. Centromere kinetochores. Centromere kinetochores. Centromere or kinetochores holds two chromatids of a chromosome. Centromere. 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 Identify the types of chromosome based on the portion of centromere shown in diagram 13. Label X, Y, and Z. X is A metacentric, B telocentric, C submetacentric, D acrocentric. X is satellite, Y secondary construction, Z centromere. A metacentric, B telocentric, C submetacentric, D acrocentric. A metacentric, B telocentric, C submetacentric, D acrocentric. And X is primary construction, secondary construction, satellite, Y secondary construction, Z centromere. X satellite, Y secondary construction, Z centromere. Dash chromosomes have centromere in the middle forming two equal arms. Metacentric. Acrocentric chromosomes have centrosome slightly away from the middle, forming one shorter arm and one long arm. False. Telocentric chromosomes have centromere at terminal end. Dash chromosomes have centromere situated close to the end, forming one extremely short and one very long arms. Acrocentric. Few chromosomes have staining or non-staining secondary construction at a constant or variable location. Non-staining constant location. Secondary construction gives the appearance of a dash. Satellite. Microbodies are not membrane bound. False. 3 examples of microbodies are peroxisome, function, photorespiration. Peroxisomes, function, photorespiration. Microbodies are present in both plants and animals. True. Flagella in prokaryotes is an extension of dash. Cell wall.